Hi, we're continuing with life science and cell biology in a nutshell. We were learning uh, so uh, about plasma membranes. So the difference between membranes of cells in eukaryotic cells versus in bacterial cells. So bacterial cells don't, which have a phospholipid membrane as well, don't have cholesterol molecules in their membranes due to the fact that they have two membranes, one being like a cell wall. So they are already rigid. They don't need a cholesterol molecule to add to the rigidity of the phospholipid cell membrane. So the eukaryotic cells have a varied um, composition of molecules in their membranes, which contribute to their own rigidity. So they have different phospholipids, they have cholesterol in different amounts, um, and in large amounts, so which adds to their rigidity. Um, because cholesterol, being a large steroid molecule with four rings, adds rigidity. So it has six carbon rings on one 5 carbon ring with a fatty acid tail, hydrocarbon tail, excuse me. So the four major phospholipids predominate in the plasma membrane of mammalian cells: phosphatidylcholine, phosphatidylethanolamine, phosphatidylserine, and sphingomyelin. These are the four major phospholipids in mammalian plasma membranes. They have different head groups, which represents um, um, so their attributes, and they are all derived from glycerol. All the lipid molecules except sphingomyelin is derived from serine. One might wonder why eukaryotic membranes contain a wide variety of phospholipids with head groups that differ in size, shape, and charge. You can begin to understand if one thinks of the membrane lipids as constituting a two-dimensional solvent for the membranes in the for the proteins, in the membrane, just as water constitutes a three-dimensional solvent for proteins in an aqueous solution. Some membrane proteins function only in the presence of specific phospholipid head groups. Just as many enzymes in aqueous solution require a particular ion for activity. So the head groups of phospholipids are important and crucial and critical in determining the proteins and molecules that interact with the head groups. Moreover, some cytosolic enzymes bind to specific lipid head groups exposed on the cytosolic face of a membrane, and are thus recruited to and concentrated at specific membrane sites. So the plasma membrane contains lipid rafts that are enriched in sphingolipids, cholesterol, and some membrane proteins. Most types of lipid molecules in cell membranes are randomly mixed in the lipid monolayer in which they reside. The van der Waals and attractive forces between neighboring fatty acid tails are not selective enough to hold groups of molecules of this sort together. For some lipid molecules, however, such as sphingolipids, which tend to have long and saturated fatty hydrocarbon chains, so singly bound hydrocarbon chains, the attractive forces can be just as strong enough to hold adjacent molecules together transiently in small microdomains, which are also called lipid rafts. thought of as transient phase separations in the fluid lipid bilayer, where sphingolipids become concentrated. The plasma membrane of animal cells is thought to contain many such tiny lipid rafts, about 70 nanometers in diameter, which are rich in both sphingolipids and cholesterol. Because the hydrocarbon chains of the lipids concentrated there are longer and straighter than the fatty acid chains of most of the membrane lipids, the rafts are thicker than other parts of the bilayer and can better accommodate certain membrane proteins, which therefore tend to accumulate there. So, a lipid raft is therefore a small specialized area in a membrane where some lipids, primarily sphingolipids and cholesterol and proteins, are concentrated. The lipid bilayer is somewhat thicker in the rafts, and certain membrane proteins accumulate there. In this way, lipid rafts are thought to help organize such proteins, either concentrating them for transport in small vesicles or to enable the proteins to function as they convert extracellular signals into intercellular ones. For the most part, lipid molecules in one monolayer of the bilayer move about independently of those in the other monolayer. In lipid rafts, however, long hydrocarbon chains of the sphingolipids in one monolayer interact with those in the other monolayer. Thus, the two monolayers in lipid raft communicate through their lipid tails. So, the asymmetry of the lipid bilayer is functionally important. The lipid compositions of the two monolayers of the lipid bilayers in many membranes are strikingly different. In the red blood cell membrane, for example, almost all of the lipid molecules that have choline, the formula being CH3, 3 nitrogen, CH2, CH2OH, hydroxy group. In their head group, phosphatidylcholine and sphingomyelin are in the outer monolayer, whereas almost all of the phospholipid molecules contain a terminal primary amino group, phosphatidylethanolalamine, phosphatidylserine, are in the inner membrane or monolayer of the membrane. So here's shown the asymmetrical distribution of phospholipids and glycolipids in the lipid bilayer of red blood cells. The colors used for phospholipid head groups. In addition, glycolipids are drawn with hexagonal polar head groups in blue. So cholesterol is thought to be distributed about equally in both monolayers. It is not shown. Therefore, there's asymmetry in the outer and inner monolayer of the membrane. The outer membrane contains phosphatidylcholine, sphingomyelin, as well. The inner contains amino group containing molecules like phosphatidylethanolamine and phosphatidylserine. And glycolipids are on the outer membrane. The negatively charged phosphatidylserine is in the inner monolayer. This is a significant difference in charge, which is created between the two halves of the bilayer. So negatively charged on the inner layer. And so lipid asymmetry is created and generated and maintained by membrane-bound phospholipid translocators. This is functionally important. Many cytosolic proteins bind to specific lipid head groups found in the cytosolic monolayer of the lipid bilayer. For example, enzyme protein kinase, PKC, activated in response to various extracellular signals, binds to cytosolic face of the plasma membrane where phosphatidylserine is concentrated and requires this negatively charged phospholipid